Hello, everyone, and welcome to today's webinar, Overcoming the Challenges of Accuracy and Detection in Molecular Assays. I am Alexis Krause of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. Now, let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Peter Holden, Senior Business Development Manager, Molecular Diagnostics, Licensing and Commercial Supply at Thermo Fisher Scientific. Peter's current role is in supporting customers who purchase Thermo Fisher Scientific products for commercial use, with a particular focus in supporting the use of molecular biology and sample preparation products for inclusion into diagnostic kits. He has over 15 years of experience in commercial sales and is responsible for ion torrent sequencing, capillary electrophoresis, real-time PCR, and molecular biology products. Prior to joining Thermo Fisher Scientific, Peter led a research group in molecular toxicology at AstraZeneca after receiving his doctorate in plant biology from the University of Edinburgh. Dr. Holden, you may now begin your presentation.
Well, good morning, good afternoon, and thank you for the introduction. In today's presentation, I will cover some of the challenges facing researchers developing a molecular diagnostic test, and I will review options that are available to overcome these challenges, including enzyme development. Lastly, I'll wrap up with the quality and manufacturing commitment from Thermo Fisher Scientific and how we can partner with you in the development of a diagnostic assay. So what is the importance of a point-of-care test? A point-of-care test allows rapid detection of analytes near the patient, which will facilitate better disease diagnosis, monitoring, and management. The disease can be diagnosed at a very early stage, enabling a quick medial decision and treatment, which leads to improved outcomes for patient health. Based on market research reports, immunoassays account for the largest market share for diagnostic testing. However, by utilizing nucleic acid amplification technologies, molecular tests are highly sensitive and specific with high throughput capability as compared to immunoassays. Many of these molecular assays are performed on portable platforms that can be run in community clinics or most remote areas of the world, setting a new bar for a true point-of-care diagnostic assay. So what is the workflow for a molecular test? It begins with the sample, the DNA and or the RNA. Today, real-time PCR is at the heart of numerous diagnostic assays, with its potential for quantification enabling assessment of DNA copy number variation, differential RNA expression levels, and pathogen load. Whereas the original PCR protocols assessed individual markers, the trend now is towards testing for multiple targets in a single assay, what we'd call a multiplex, which not only allows for high throughput testing, but also reduces cost and the time taken to complete the assay. For our presentation, we'll focus on the amplification method used in real-time PCR. So there are many challenges to overcome when preparing purified target material for qPCR. One such challenge is the time to prepare material, which makes it susceptible to environmental and sample-borne inhibitors. To detect the underlying molecular changes in a diagnostic test, a variety of specimen types are needed, which may include cells from buccal swabs, saliva, blood, frozen or paraffin-embedded tissue, stool samples, and many other types. However, many of these samples have inhibitory factors, and the presence of these factors, such as hemin in blood, can result in the inhibition of the amplification reaction, resulting in false negative results. Other challenges include carryover of enzyme inhibitors from extraction reagents and poor yield, which is further complicated by low amounts of intact target DNA in the purified sample. For example, blood is a common sample type. Cancer blood tests work by screening for cell-free DNA released by dying tumor cells. These tests, known as liquid biopsies, are showing promise, but not only for the detecting cancer DNA, but also monitoring treatment progress. Tumor DNA makes only a fraction of the total of CF DNA. Thus, to avoid false negatives, minute amounts need to be detected, and significant amounts of DNA needs to be obtained to avoid biases and artifacts. This is further hindered by inhibitors present in blood itself or coming from blood stabilizers or purification. As you develop a molecular assay, the choices you select for DNA, RNA extraction are interconnected to amplification steps, especially in the enzyme choice. Traditional enzymes such as Taq, DNA polymerase, which are less tolerant of inhibitors, will require a cleaner sample. Therefore, more investment into the DNA-RNA extraction step is needed. However, if better amplification enzymes are available that are tolerant of inhibitors, then the requirement for highly purified targets is less critical. Not only do better amplification enzymes need to be tolerant of inhibitors, but they can also offer improvements in other areas within your molecular assay. These could be areas such as accuracy, early detection with a faster turnaround time, fewer steps or processes for ease of use for the operator, room temperature stable formats for ease of storage, multiplexing, as we've mentioned, but lastly, probably the reliable and robustness for a variety of sample types is critical in developing your molecular assay. In this section, I will review the key properties of a preferred enzyme in detail, which will include the following: multiplexing, to increase the number of target per reaction and conserve precious samples; processivity, which is the greater affinity for the template; inhibitor resistance, and working with challenging samples. Another area is thermostability, room temperature stable variants, as well as thermostability to alleviate elements such as secondary structure. Specificity and speed are also critical. Speed, as measured by the number of nucleotides incorporated per second, provides higher throughput and faster time to diagnosis. Multiplex PCR technology allows simultaneous detection of multiple nucleic acid targets in a single reaction as compared to a single plex reaction. The gel data shown here is an example of hot start platinum-2 DNA polymerasing, multiplexing 15 targets in a single PCR reaction. The advantage of multiplex PCR in a diagnostic test is that it increases the number of targets in a single reaction and reduces the sample requirement and eventually and ultimately conserves precious material. The next key attribute of a better enzyme is processivity, which is the ability of an enzyme to catalyze consecutive reactions without releasing its substrate, as shown in this illustration. Highly processive enzymes incorporate a higher number of nucleotides per binding event, and this results in much longer applicons. Due to a high affinity for the template, these highly processive enzymes are more tolerant of inhibitors, as shown here in this figure, and in particular in figure A. Inhibitors like heparin or formalin have a minimal effect on a highly processive enzyme, such as SuperScript 4 reverse transcriptase in the cDNA synthesis step, as compared to enzymes with low processivity. Figure B shows that a highly processive enzyme can synthesize cDNA from degraded RNA more efficiently than standard enzymes. If RNA is used as a starting material, then a high level of RNA secondary structure can cause a problem for the cDNA synthesis step. An attribute of a better enzyme is one to perform at higher temperature where secondary structure is alleviated, as seen here by this figure in this illustration. This illustration shows that RNA templates of varying lengths were reverse transcribed at different temperatures using a highly thermostable engineered MMLV reverse transcriptase. The results indicate thermostability of the enzyme with 100% activity even at 56.4 degrees centigrade. Specificity of an enzyme is another property important to a diagnostic assay because of the precise interaction of the target template with the enzyme. One feature is to use a hot start DNA polymerase enzyme, which will only be activated at a specific temperature, which is convenient if requiring room temperature setup. In this gel data, a PCR reaction was performed after setup at room temperature using platinum 2-Tac DNA polymerase. Here we can see that there is no non-specific amplification even after 72 hours incubation at room temperature. I must add to this figure, PCR reactions were prepared and incubated at room temperature for 0, 24, and 72 hours before loading into a thermocycler. Highly specific amplification of a 2 kilobase fragment from human genomic DNA was observed even after 72 hours after room temperature setup, demonstrating the power of hot start DNA polymerases for large-scale experiments. In figure A, as you can see on this slide, we have another gel which shows the absence of a non-specific amplicon with hot start DNA polymerase. In addition, in figure B, High specificity can be enhanced with thermostable enzymes as seen at higher temperatures. These are PCR results from non-hot start versus hot start DNA polymerases. Please note the improved yields of the desired amplicon and the lack of non-specific amplification with a hot start DNA polymerase. Reverse transcription at an elevated temperature enhances PCR specificity. mRNA for 4 different genes was reverse transcribed at different temperatures using oligo-DT or gene-specific primers of varying length. Reverse transcription at 55 degrees centigrade generated higher specificity of the desired targets in the RT-PCR reaction. Enzyme speed is important since it will improve time to result in a molecular test. The example shown here is a 15-second PCR amplification as compared to a typical amplification that might take 30 minutes or more. So, do you need all these enzyme properties for your molecular assay? As you develop your assay and review these different attributes, you must understand there has to be a trade-off. For example, do you use a better enzyme to overcome suboptimal samples against or versus a lengthy sample processing step to obtain your purified target? So that has to be taken into consideration.
Thank you, Peter. And now that takes us to our first poll question. We'd love to hear your input here. True or false? Hot start DNA polymerase utilizes an antibody or chemical modification so that the polymerase becomes active only at the high temperature of the denaturation step. True or false? Simply click on that answer. Give you another moment to respond. Okay, we will continue with Peter.
In the next section, I will present information on advances made in enzyme development by Thermo Fisher Scientific, which will help you make better choices in assay development. We use several approaches in developing a better enzyme. First, start with the protein design to obtain an enzyme with a desired phenotype. Second, examine the buffer formulation and optimize for specific applications. And then lastly, implement single-use technology to manufacture enzymes that contain with contaminating DNA below the limit of detection. So let's take a look at these approaches in more detail. The classic approach to protein design is to isolate from a natural source, such as Taq polymerase from Thermus aquaticus that lives in hot springs and hydrothermal vents. However, this method relies heavily on luck to obtain the desired feature. The second approach is by rational design. An example of this is the SuperScript 2 reverse transcriptase, which is a point mutation in a gene encoding the Maloney-Murine leukemia virus, MMLV, reverse transcriptase. This method requires knowledge of the domain or the amino acids of the enzyme. The last approach is protein evolution, also referred to as directed evolution, This method mimics the process of natural selection to steer proteins towards a desired feature with no prior knowledge of protein structure or function needed. So this seems quite desirable. How does it actually work? The process starts with a gene encoding for a wild type or unmodified enzyme of interest. Random variation is introduced into the gene through a process of mutagenesis, generating a library of millions of genes each encoding for a unique enzyme variant. Functional selection pressure using, for example, higher temperatures is then applied to the library where only the genes that coded for the highest performing enzymes actually survive. This process of random mutation selection is repeated until the desired enzyme function evolves. Directed evolution enables us to engineer improvement to enzyme functions such as processivity, thermostability, and more tolerance to inhibitors. These evolved enzymes are different from other engineered enzymes because they have been optimized for specific functions. To further the development of better enzymes, we can also utilize gene fusion technology to insert a protein domain with a desired feature into the novel enzyme. One of the Thermo Fisher enzymes, fusion DNA polymerase, is an example of gene fusion, which brings together a processivity enhancing domain with a pyrococcus-like, also known as PFU, enzyme. So utilizing a combination of methods, Thermo Fisher Scientific has developed several new generations of enzymes, such as platinum-2 Taq DNA polymerase and SuperScript 4 reverse transcriptase that can help overcome some of the challenges you face in developing molecular diagnostic assays. Platinum-2 Taq hot start DNA polymerase was designed by two innovative technologies. The polymerase was engineered by protein evolution for higher speed and resistance to PCR inhibitors, while an innovative PCR buffer enabled universal annealing temperature. In the top panel, the Platinum-2 Taq shows higher tolerance to common PCR inhibitors as compared to other commercially available DNA polymerases. In the bottom panel, the ability of Platinum-2 Taq for faster DNA synthesis or fast cycling can result in an overall reduction in PCR time. These features are critical to any diagnostic assay, especially when working with suboptimal sample types and when requiring a fast turnaround time in detection. Similar to Platinum-2 Taq, SuperScript 4 reverse transcriptase was also designed by protein evolution for speed and resistance to inhibitors and an optimized buffer formulation. In the top panel, the graph demonstrates that SuperScript 4 RT resulted in the lowest CT value when compared to other commercially available RTs. In addition, the reaction time for SuperScript 4 is 10 minutes compared to, say, 30 to 60 minutes for the other reverse transcriptases. In the lower panel, inhibitors that can interfere with cDNA synthesis, such as hemin, has very little effect on the performance of SuperScript 4 reverse transcriptase. Thus far, I've covered protein designs and optimized buffer formulation for the development of better enzymes. In the next section, I would like to present the use of single-use technology for enzyme manufacturing. How does manufacturing impact the performance of better enzymes? From various publications, it is known that commercially available Taq polymerases contain various strains of bacterial DNA. As you can see here, this is quite a detailed slide, but gives a strong illustration of the number of species that can be found within DNA. These enzymes are typically contaminated with bacterial DNA and other contaminants during manufacturing. Most of these enzymes are recombinant protein expressed in E. coli. Therefore, it's not surprising that they'll contain some bacterial DNA, as seen in this graph of a qPCR assay using no template control here in the bottom left-hand corner. An uncontrolled DNA contamination in Taq polymerase used in a molecular assay can compromise both sensitivity and specificity of an assay. Using a closed single-use system, as we call SUS, where disposable single-use components such as filters, bags, and columns are connected by sterile single-use tubes for every stage of the production. The closed systems ensure that an entire process is never exposed to the surrounding environment or the operator. The product from this manufacturing innovation is a DNA-free DNA polymerase. The top panel shows two graphs of qPCR assays performed using the DNA-free platinum Taq versus commercially available DNA polymerases. And both are tested for the absence of signal and a no template control. No amplification was observed in DNA-free platinum Taq, whereas presence of contaminating DNA and other enzymes can lower analytical sensitivity of a molecular assay, since the signal of the contaminating DNA can interfere with detection of low-copy DNA targets. The tight quality control for DNA-free enzymes provides an increased confidence in the sensitivity, specificity, and reproducibility of your molecular assay, especially for low-copy DNA target detection. As more point-of-care tests are being developed, so is the demand for room temperature storage and stability of assays. Unfortunately, the majority of commercially available enzymes on the market are incompatible due to the presence of glycerol in the enzyme buffer. Our solution is a Lyo-Ready analog that contains a glycerol level below 0.5% and still retains the functions and performance of enzymes containing glycerol. So with considerable experience in identifying and optimizing buffer formulations and lyophilizing, we offer a large portfolio of Lyo-Ready enzymes such as SuperScript 4 reverse transcriptase that can meet the highest standards of performance required in your molecular assay. You can see from the figure that this enzyme performs the same in various formats, i.e. in Lyo-Ready format, lyophilized, reconstituted, and also the standard glycerol formats. So this data is showing the effect of buffer pH on thermostability. The protein thermal shift assay was used to screen for the effect of various buffer concentrations on REC-A thermal stability. Data from the protein thermal shift software shows the Boltzmann upper panel and derivative lower panel melt profile of buffer A, pH 5.5, buffer B at pH 6, C at 7, and D at 7.5. The data was collected at 1 degree centigrade intervals from 25 degrees C through to 99 degrees centigrade on Applied Biosystems Step 1 Plus real-time PCR machine, and analyzed with the protein thermal shift software.
And that brings us to our next poll. Would you like to be contacted by Thermo Fisher's business development manager to get more assistance for your research? Yes or no? Simply click your answer. And again, that question is, would you like to be contacted by our business development manager to get more assistance for your research? Yes or no? I'll give you another moment to choose your response. Okay, to part 3.
These new generation of enzymes, which will be customized to meet your individual specification, are manufactured in our Vilnius Biosciences Center of Excellence. The site has over 40 years of experience, expertise in enzyme development and reagent manufacturing, and the facility employs state-of-the-art equipment and is capable of small-scale to multi-liter volumes of reagent production. The capacity and flexibility of our production facility enables us to respond quickly to any throughput requirements. Thermo Fisher Scientific understands the importance of adhering to ISO quality standards when developing and supplying reagents for molecular assays. All production work is performed in accordance with ISO management systems, which means we are committed to being a reliable partner that has a deep understanding of our partners and our customers' needs. Our commitment to continuous improvement has been recognized by Thermo Fisher being awarded the most prestigious Shingo Prize, which recognizes organizations for operational excellence. So this is my final slide. And I just want to encourage and thank you for listening to this webinar. And also just to say, you know, if you have an interest in developing a molecular assay, and you have an interest in the products that we can offer and the attributes these products have, then we encourage you to get in touch with us at Thermo Fisher. Our business development partners offer a large amount of experience and a vast technological depth that we can impart on you, and we can draw in expertise from various teams within the company, especially within our R&D function, to help you develop your assays and suggest alternatives, or even suggest alterations in your current setup.
And that brings us to our final poll. Do you have plans to develop nucleic acid development assay? If so, is it 3 to 5 months, 6 to 12 months, or no, you do not have any plans? Simply click your answer that fits on your screen. Again, the question is, do you have any plans to develop nucleic acid development assay? 3 to 5 months, 6 to 12 months, or no plans? I'll give you a moment to respond. Okay, thank you for your participation.
So again, yeah, just please get in touch if you're interested and you have interest in the topics that have been discussed during this webinar. So I will now close, but I would maybe like to call on you for any questions. If you have any questions you'd like to ask, more than happy to answer them at this particular point. But thank you for listening, and I'll take some questions now.
Thank you, Dr. Holden, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Answer Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. So let's get started. Our first question is, thank you for the informative webinar. My lab develops molecular diagnostics for NTDs such as malaria. Our objectives are to produce MDXs that are sensitive and specific, as well as field-friendly. I would like to know if samples are available for my Lyo-Ready enzymes. I would like to determine if these enzymes may aid us in our goals.
Well, thank you. Thank you for your interest. Great question. Yeah, we offer a wide range of DNA polymerases and reverse transcriptase in Lyo-Ready format, and we would be glad to offer samples for testing. For sure, we can send some to you. But for more details of our Lyo-Ready enzymes offering, I would suggest that you visit our website, thermofisher.com/Lyo-Ready enzymes. That's one word. So that's visiting thermofisher.com and the /Lyo-Ready enzymes. And leave your contact information and on the inquiry form, and one of our business development managers will contact you and set you up.
And it looks like we have time for one more question, Dr. Holden. In your presentation, you had mentioned one of the enzyme properties as multiplexing. I am working on multiplexing diagnostics assays and want to know if Thermo Fisher has any recommendation for DNA polymerases.
Well, thank you for your question. In fact, we have platinum 2-tack hot start DNA polymerase that I would recommend you checked out. This enzyme is one of our new generation of enzymes combining both protein design and unique buffer composition that I alluded to in the presentation, and that allows for a universal annealing temperature. Platinum 2-tack hot start can be used simultaneous amplification of many targets in a single tube using multiple pairs of primers. However, I do recommend verifying the performance of each primer pair in a singleplex PCR before starting the multiplex reaction. And don't forget that primer design parameter is still critical for successful multiplex PCR. Again, for more information about this enzyme, please visit our website. Oh, I must add to that our R&D teams are always ready to help in answering specific questions such as these and providing consultation relating to the use of our enzymes. Thank you.
Thank you, Dr. Holden. Do you have any final comments for our audience?
No, just thank you for listening.
Thank you again, Dr. Peter Holden, for your time today and your important research. We would also like to thank Labroots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information that you provided at the time of registration. This webcast can be viewed on demand through July of 2019. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.